Good afternoon, everybody, and welcome to the Lunchtime Discovery Series. I'm your host, Chris Smith, and I work here at the North Carolina Museum of Natural Sciences. It's good to be with everybody again for another edition of our lecture program. We meet here every Wednesday at noon to meet some interesting people and learn interesting things that are happening, generally speaking, around the state of North Carolina in science, nature, art, education, and more. This program is brought to you as a broadcast service by us here at the Museum of Natural Sciences in downtown Raleigh, but is organized and put on by the folks with the North Carolina Office of Environmental Education. They do a lot of promotional support for this program too, so we have a great partnership to bring what it really is an amazing program out there to the world every single Wednesday at noon Eastern. Make sure that you mark your calendars and tune in. And make sure that you go back and look at some of the previous videos as well. There's a great archive of lunchtime discovery programs right here on the museum's YouTube channel that you can take advantage of. Find ones, give it a scroll that you think are interesting, and give those a watch. There's some really good people talking about really cool stuff happening across the state and sometimes even around the world. So great things happening right here with this program every week. And today will be no different. Today we actually have 3 guest speakers who are going to be talking with us today about work that they've been doing out on the coast. I'll remind everybody that as we go through the program and as we learn about today's topic and meet our guest speakers, leave your questions and comments for our guest speakers in the chat if you're watching on YouTube, or in the comments if you're watching on Facebook. I've got those pulled up. So I'll be moderating, and when we get to our audience Q&A segment at the end of the presentations, I'll be looking to take your questions and share them with our guests. So as we go through, make sure you're queuing up those thoughts and questions so that we can have a great discussion about today's topic. Okay, I think it's time to actually bring on the first guest speaker that we'll hear from for today's program. Everybody, I want you to meet Dr. Carol Price. Dr. Price is the Conservation Research Coordinator for North Carolina Aquariums. Of course, the aquariums are part of the Department of Natural and Cultural Resources, just like us. So it's great that we've got a little bit of sister agency stuff, sister department stuff going on here with the museum. But Carol is also an instructor at the NC State Center for Marine Sciences and Technology. Carol, welcome to the program.
Thank you so much, Chris. I'm really excited to be here today and share a little bit of the research that I do as part of the Aquariums Conservation Program. And it's a nice tie-in because my co-presenters today are students at NC State University. We'll meet them a little bit later in the show. So thank you so much for the invitation. And also thank you to all of the people out there who took your lunchtime today to tune in and learn a little bit about a very little known creature, but one that's very near and dear to my heart. So I want to tell you a little bit about the crystal skipper today. So I'm going to go ahead and start sharing my screen. All right. So the title of this talk is Skippers in the Dunes, and you can see where I work and all of that, but really this is a really important time to tell you sort of the real subtitle of my talk, which is, and how did a fish biologist, marine scientist end up studying butterflies and dune vegetation? So disclaimer, I am actually not an entomologist or a botanist, but somehow I have become the program lead for this field and research project. And so I'm going to sort of be telling you 2 stories today, one about the skipper, but 2 just about how if you take some chances, you get to maybe get outside of your normal lane of operation and do something that's a little bit new and exciting. And just how do you actually go about doing something like that? And there we go. So going to start with just a basic background on the crystal skipper. The thing that makes this butterfly so unique is not necessarily how it looks. It's a pretty small brown butterfly, kind of plain. It's got beautiful little crystal spots on its wings, which is part of how it got its name. It's about the size of a quarter. And so it's not the flashiest, most beautiful butterfly out there. but it's really special because it only occurs, it's only found in a really small part of North Carolina, specifically out in the middle part of the coast where I live, near Morehead City. And its range is bounded on the east side by Fort Macon State Park and on the west end by Hammocks Beach State Park. Just kind of remember, our coastline actually sits more east to west than north to south in this part of North Carolina. So that's why it's an east-west. So it's only found on this little island stretch that's not very much area, about 8,000 acres or so. And because, as we know, barrier islands are really popular places for people to live and to come as tourists, there's a lot of interaction between this species and human activities and human structures like houses and condos and parking lots. And to make this even more restricted for this species, it doesn't occur all over the barrier island. It's only going to be found in a really small swath of habitat that's behind the primary dune line. So if you're looking at the ocean and you have that first sand dune and it's where the sea oats are growing, right about the space on the backside where the sea oats stop, all the way back to where the maritime forest begins, there's this kind of habitat, these sort of swales, dune swales behind the primary dune line. It's sort of prairie looking, it's very inhospitable, it's very dry. You have this sort of grassy expanse out there, not a lot of big plants or showy plants, a few flowering plants from time to time, but it's very dry, it's very hot, there's prickly pears, and this is where this little butterfly calls home. And the reason for that is because of one specific plant called Seaside little bluestem, which is what you see here. And this plant is important for the skipper because for now, it's the only plant that this butterfly is known to lay its eggs on. And fortunately for the butterfly, this is not a rare species of plant. This is a common grassy plant that's found throughout most of the dunes, especially where they're healthy and we haven't had a lot of invasive species showing up like centipede grass. And so it's also important because once the eggs hatch, this plant is the food source for those caterpillars. So those caterpillars have got to hatch out and just munch and munch and munch and munch before they can form into adults. And that nutrition is going to help them through metamorphosis and then into the mating and egg laying stages. So the crystal skipper has a life cycle very much like other butterflies. They mate, they lay eggs, as you can see here on Seaside little bluestem. Those caterpillars emerge and hatch and start to munch through that vegetation. They form a chrysalis and then they hatch into or metamorphose to their final transition to an adult butterfly. The butterflies are only around, a butterfly only lives a couple of weeks. And so in those few days, it has to find a mate and then lay some more eggs. Interestingly, for this species, there's 2 flight periods every year. What I mean by that is in about mid-April, butterflies emerge, mate, lay eggs, and by about mid-July, those butterflies hatch out or emerge and go through this same process again and lay their eggs. But those eggs stay dormant through the winter and hatch back out the following mid-April. So if you're in the dunes in April and you're seeing skippers, what you're actually seeing are the grandchildren of the April skippers that you saw the year before. So that means that there's 2 flight periods, and that'll be important a little bit later when Katie and Maggie are talking. And as for many other species, the crystal skipper is facing habitat loss. Here are 2 photographs of the same little stretch of beach in Indian Beach, which is about halfway down Bogue banks. And you can see between the 1960s and today, there's been significant alterations to the habitat, a lot of human development, and especially in the place that the skippers rely on, which is that on that ocean facing side. So if you look at the bottom of each photograph is the ocean side, at the top of each photograph is the sound side, and the butterflies don't use that sound side habitat. So it's just that real thin stretch along the ocean there that they need. But it's also where a lot of beautiful homes are, large homes, condos, parking lots, businesses are also found there. So a lot of opportunities there for interactions. And sadly, unfortunately, in many cases, that leads to habitat degradation. That is a combination of direct loss of habitat because a structure has been placed on top of it. It can also be where we have damage to the dunes because of traffic through the dunes and those sorts of things. It can also be, as I mentioned before, when people plant invasive or ornamental species, those can invade into the dunes and outcompete the native dune vegetation, including the seaside little bluestem and nectar plants that the skipper might rely on. So, so this meant that for this butterfly to persist, there were a lot of questions that needed to be answered about how or even if that was possible. And this is what led my colleague, Dr. Allison Leidner, to select this species to study for her PhD work in the early 2000s. And so she was here and spent 5 or 6 years studying the crystal skipper and its population and its habitat. And she really came up with 2 really important conclusions from her dissertation work. The first is this conservation model that I'll talk about on the next slide. But then the other really important thing that she did is that she was able to collect enough samples of this butterfly and was able to arrange for the genetic analysis that needed to be done to confirm that this was not just a subspecies or a species that was found on the mainland, but in fact that it was a new species, which is important to have that designated. So that just happened within the last 5 years that species designation happened. And that really opens it up for us to be able to study this population as its own species in its own right. So let's talk a little bit about what I mean by this habitat stepping stone model. So as I mentioned, the Fort Macon State Park is at that east end of the habitat, and that's what you're seeing in that big yellow box. There's a lot of beautiful dune habitat there. Historically, lots and lots of crystal skippers are found there because it's a park. It's nice and protected. So the skippers are able to persist there. But all the way down to Fort Hammock Beach State Park is a really different story. What we have instead is sort of patches of suitable habitat, which are those yellow circles that you see here. So interspersed between the built environment are these small patches, either at other public lands or some county areas, some private houses, and so forth, where there is suitable habitat and where crystal skippers have historically been found. And importantly, what Dr. Leidner was able to establish is that the butterflies do not need a continuous corridor of suitable habitat to be able to move up and down the beach and sustain their population. Rather, they were able to move between these strong populations at the parks and use these others as sort of stepping stones and fly between them and find those patches. And that's a really important finding because it means that it is likely that with proper management and conservation that this butterfly can persist in this environment, as long as we make sure that on either end those populations are good and that we have stepping stones in between that are also protected. So how did this partnership start? I started working for the aquariums about 5 years ago, but well before that, I had heard about the crystal skipper. So this little butterfly is actually sort of a local legend. Everybody sort of knew that there was this student working on this butterfly. And so I had heard about all of this and was interested in it, but I had never really gotten sort of the, deep into it. About that same time, Dr. Leidner was leaving to take a job in Washington, D.C. And I'm not exactly sure how we connected, but somehow we got together. And as so many stories start, this one started with a walk on the beach in 2017. So we met at a couple of her field sites. She told me about the butterfly, and she told me about some of the work that still needed to be done, some next steps following up on her dissertation work that would really help continue the work for this species. And I told her, I'm like, I'm a fish biologist, I don't know about this. And she said, you've got this. It just needs a champion to help pull the experts together and just keep things moving along. And so, because I knew that part of my job was going to be working with student interns who needed field research projects during their semester down at the coast, I made an agreement that if I could find a good student who was willing to do the work, I would give it a try. But she couldn't just go to DC and abandon me. She had to stay on my team, and she agreed to that. And so I got very, very lucky. I was able to work with a student, Ian Grace, who is now a graduate student at NC State University. And he and I agreed to work together to figure out how a couple of marine biologists do butterfly work. And so both of us took a deep breath, threw in a dash of courage, and moved forward. So the first thing that we needed to do was to figure out a good project for him. So what Allison was interested in looking at was, what is the status of the vegetation community, the dune plant community, at all of these locations where we knew butterflies had been found historically. And so Ian selected 8 sites along Bogue banks to conduct his plant surveys. And those are where you can see those push pins there. And these were sites that were sent to us by Allison, and then a couple of others that we selected. And at each of those locations, he delineated in pink the sampling site. And then he randomly chose 20 points where he would then conduct vegetation surveys. So neither of us being botanists, we decided that we needed to keep the plant surveys pretty simple. And so we, Ian, I'm going to give Ian credit for this because he really did all the research on the methodology here. But we decided to do 1 meter plot square assessments. We were going to do abundance, so not trying to identify necessarily everything or count everything, but just to kind of get an idea of how abundant the major dune plants might be. We were especially interested in Seaside little Bluestem for this, to make sure that there was good coverage of that. This is sort of what his data sheets look like. So at the top, you can see SLB is Seaside little Bluestem. We were also interested in how much bare ground might be found within each quadrat. And then we picked out a few other plants which is a combination of plants that we could identify and plants that we also felt might be important based on Allison's work for the skipper. And so at each of his sites, for each of the 20 points, this was the data that he collected. And at the end of his semester, he was able to put together a graph that showed, characterized the relative sort of coverage of these plants. And so what you can see is in the light blue is the Seaside little Bluestem, and the orange is the bare ground. Those are sort of the 2 main points to take away from this. And then a few other plants there in the upper parts of those bars. And so what we also had was some seeds, Seaside little Bluestem seeds that had been collected by previous researchers for the purposes of doing some restoration work. And so we took this opportunity to use the data that he had collected to identify 2 locations that had a combination of low Seaside little Bluestem and high bare ground, and chose those as our restoration sites to add a little bit more vegetation to those areas for the skipper. And so over the summer, Ian was able to continue this work with funding that he got through Dr. Nick Haddad to do plantings at those 2 sites. So we pulled together a crew of volunteers. Up in the brown t-shirt with the blonde hair, that's Ian. So he organized all of this. That's me there in the blue shirt, but you can see that we're taking those plugs of Seaside little Bluestem and planting those in the ground. We also recruited the Master Gardener group from locally here in Carteret County, and they were a big help. And then a few more students, including my 14-year-old son there, who doesn't look super thrilled to be on this expedition, but he was very helpful nonetheless. And so we went out one day and we put all of these plugs in the ground at these two sites. And we're really excited about having done this project. And then a few months later, you might have heard, there was a hurricane. Hurricane Florence came through later that year. And as many of you know, it had an enormous impact on the local barrier islands here at Bogue Banks. Specifically, there was a lot of overwash from the salt water as the sea just sort of came in over those dune lines. And not only did it wash up very high, but it also sat in some places for a very long time. That salty, briny water. It sat for a long time, and some areas were just very scoured out from the impacts from that storm. So I was a little worried about what that meant for the Bluestem as well as the skipper populations along the island. So about this time, I was very fortunate to get in touch with Gloria Putnam. She is the program lead for the Coastal Landscapes Initiative that's operated out of North Carolina Sea Grants. And the purpose, you can read there for yourself what their goals are. But she really saw this as an opportunity and a need, kind of matching up here with her program and what happened with our habitat. And so she was able to write a grant for a little bit of funding to help support another restoration project in these areas where there was a lot of scour and potential habitat loss for the skipper. So the first thing we needed to do, though, was figure out where that loss of habitat was the most intense. And so in April of 2019, we went back out to those exact same sites and did just redid the exact same surveys that Ian had done the year before. And you can see, and here I've only included the Bluestem and bare ground data, but what you can see is that at almost every single site, the amount of bare ground increased significantly, while in some places, the amount of Seaside little Bluestem decreased. And so based on that, we knew we would be able to do some restoration. And so based on these results, we selected 2 sites to focus our restoration work on our next round. However, we didn't have any more seeds. So the first thing we had to do was go out and get some more seeds. And to do that, we connected with Mike Kuntz, who's up here in the top right. He is a botanist, ecologist, plant ecologist at the North Carolina Botanical Gardens, and is very, very knowledgeable about dune plants, as well as how to raise them. And so he came down with some of his staff and students. We were joined by several other volunteers. For example, Ben Fleming, who is one of the park rangers at the state park, Fort Macon State Park. He and the superintendent, Randy Newman, are strong supporters of the crystal skipper, very proud to have them there. And so they took us out. So Ben took us out in the dunes and showed us where we could collect seeds. And so our team spent the day collecting seeds, which you can see in the bottom left there are these sort of fuzzy, fluffy little seeds that we collected as many as we could. And then we sent them back with Mike to his greenhouse setup so that they could grow out those seeds up there and have them ready for us to do another restoration planting. In addition to Bluestem, because we had Mike there and because he's really good at identifying plants, we were also able to collect seeds from other plants that are in the native dune plants that we think might be potential nectar sources for the butterfly after they emerge. And so we also collected seeds from the plants that you see here. We did not collect seeds from a couple of other plants that are also known to be important nectar sources for crystal skippers, but hopefully we can maybe do that at a later date. So by October 2020, those plants were all healthy, as you can see there in the top picture. Very nice, healthy seaside little bluestem. However, as if one natural disaster wasn't enough, we also had to throw in a pandemic just to make it really exciting and hard to do restoration work. So we had to get special permission from our many agencies. And because this was all work outside, we were able to get that exemption to travel and do this work. Notice this was before the vaccine was available, so we all had to wear masks all the time. So we were really, really careful to make sure that everybody stayed safe. And we did restoration planting at 2 places. One is the location on Atlantic Beach, a skate park, near a skate park, and the other location was at a private property on Emerald Isle. So there you can see our crew. That includes folks from the botanical gardens, aquarium staff, volunteers, and Gloria Putnam from Sea Grant. We also had some master gardeners and other folks that came out to help. In addition to those 2 restoration sites, we also planted at 2 demonstration gardens or demonstration sites. One is at the Salter Path Beach access, and we wanted to put some plants in places where people could see the plants, and we can put up signs and explain and have some education information there for the public about this butterfly and the habitat that it relies on. So in addition to the Salter Path location, we also were able to put in a demonstration garden at Fort Macon State Park. We finished that up in early 2021. Mr. Fleming had to remove some invasive plant species before we could replant with these native species. And then my friend Georgia Minnick designed a really beautiful graphic that we could put up on the boardwalk that explained what the crystal skipper was, why it is so special, and then a little bit about the plant community that it relies on. Also in 2021, I realized that with this sort of work behind us, I could move on to some next steps maybe for the skipper. It had been a couple of years. I'd had great movement going forward with all these efforts, lots and lots of people helping out. And so I felt like it was also time to take on a new group of undergrad interns to help with some additional new priorities for this project. So specifically, we were interested in expanding that list of plants that might be serving as nectar plants. So what I needed was students to work on coming up with a list of plants that are flowering in the dunes during those 2 flight periods, trying to go out and actually photo document skippers on those plants so that we knew that of the plants that were flowering out there, which ones were the most important for the skipper. And then finally, we also wanted to conduct some really good population surveys again. So since Allison had left, Heather, who works with Dr. Nick Haddad as a technician, had been going out a couple days during each flight period over the past few years and doing point counts at some of these locations. But it had been really quite some time before, since a more rigorous monitoring program had been put in place. And so we felt like that was also a really important thing to do. And so I got very, very lucky again with my interns. I was able to get 2 wonderful, wonderful students to work on this. So let me introduce very quickly Katie Barnes on the left, who was sort of the lead skipper intern for this project during both the semester that she was here at CMAST, but then also these 2 ladies applied for grants and got them to stay through the summer. So both Katie and Maggie there on the right were able to work really hard on achieving those goals that I just laid out for you in the previous slide. And so at this point, I'm going to turn the talk over to them and let them share their story of what they got to do to help the crystal skipper.
So thank you, Dr. Price. Let me just share my screen really quick. So my name is Katie Barnes, and I'm a senior at NC State, majoring in plant biology. And I'm also minoring in marine science and animal science.
My name is Maggie Hart. I'm also a senior at State. I'm studying environmental science, and I'm minoring in marine science and biology.
So we were two of Dr. Price's interns this summer, and the next portion of this presentation is going to focus on our experiences this past spring and summer. The first portion we're going to discuss is vegetation surveys, and I will do that portion, and Maggie will wrap up with skipper surveys. So we performed our vegetation surveys using a set of quadrats. So now I will go over some Quadrat Constructing 101. This slide essentially details how we constructed our quadrats. We used PVC pipes to connect 3 squares to create nested quadrats. First, we cut the PVC pipes to appropriate lengths and then connected them with PVC connectors, or when they inevitably kept falling apart in the field, we finally had to super glue them together. So our first and smallest quadrats, as you can see from the picture here, the first smallest one is 25 by 25 centimeters, and the second one was 1 by 1 meter, and the third was 2 by 2 meters. This would allow us to determine the frequency of species within each plot. So once we finished our quadrats, it was time to find points to sample. We used Ian's data points so that we could resample locations he did in 2018. However, some locations were new and we had to use a randomized approach to determine the new sampling sites within our area of interest. So here's a map of Fort Macon and the points that we sampled at these 2 sites. And Fort Macon is one of the best known places that we see skippers, and it's on the farthest east end of their range. And this is another location. This is Bear Island, which is a part of Hammocks Beach State Park. And this area is the farthest west of the skipper's range. However, we also sampled areas in Atlantic Beach, Salter Path, Indian Beach, and Emerald Isle. So once we set up the correct coordinates in the GPS, we carried our quadrats through the dunes and randomly dropped the quadrats at our sites. Then we counted all species that we saw in each of these quadrats and determined percent cover to determine abundance in quadrat 2, the 1 by 1 meter square. This was to ensure that we could compare Ian's vegetation data to ours in a way that was the same. This took a lot of practice to be able to identify the plants in the field, but once we got the hang of it, we could go into the dunes and jam to some Taylor Swift while counting our plants. So after a long month's work of identifying and counting plants in the field, we finally finished and were able to create results of our data to determine patterns and trends. This graph right here is a summary of the species we saw at each of our locations and how many we saw. We can conclude from this data that overall the sites were with the greatest density and total plants within quadrats was at Fort Macon. We also see that the skate park site in Atlantic Beach and Bear Island sites have the fewest average number of plants recorded across each of our 10 coordinates we sampled. So as you can see, the large green portions right here, this represents Seaside little bluestem. And as Dr. Price mentioned, this plant is of vital importance to the skippers as it's the only host plant that we've seen and know they cooperate with. Additionally, we saw a lot of Seaside little bluestems at all the sites, except for our new site, Trinity Center. Additionally, the large blue sections on the graphs here, those represent some other plants that we saw, including sea oats. And some of the other kind of big portions that we saw, those included Seaside little bluestem, like I mentioned, sea oats, dune pennywort, panic grass, and prickly pears. So this second graph here was our percent cover graph, and we used this to create estimates about the distribution of species on the 1 by 1 meter square plot. The large blue sections on this plot, They represent the bare sand, and the teal represents Seaside Little Bluestem, and the lighter green right here represents sea oats. So this data was collected to get a comparison of what vegetation was like in 2018 when Ian performed his surveys. So now in this graph, we have a comparison. We can see that there's a lot more bare sand from when I performed my surveys. And we can actually see less Seaside Little Bluestem as displayed by the blue bars. Therefore, it's critically important that we keep planting the Seaside Little Bluestem since it is the only known host species of the crystal skipper. Changes between these 2 years can be attributed to a number of factors, including Hurricane Dorian and other hurricanes that we've experienced that could have damaged the dunes. So what's next? This semester, we have worked on calculating diversity indices from the data that we collected this past spring and summer. We have worked with a conservation ecologist, Mike Kuntz, to help us determine the most appropriate methods for calculating diversity at our locations. Currently, we are working on calculating the Shannon Diversity Index, which will allow us to characterize species diversity across a community. These calculations will eventually allow us to correlate vegetation abundance with skipper abundance in tracking species diversity over time. So now I'm going to hand it over to Maggie Hart, who will be discussing the crystal skipper surveys that we did.
Thanks, Katie. So I'm going to talk about crystal skipper surveys, the methodology behind them, and some of the results from our fieldwork this summer. So like Dr. Price mentioned, the crystal skipper has 2 flight periods, one from mid-April to mid-May, and the other towards the end of the summer in mid-July to mid-August. So as the time kind of neared for both of these flight periods, Katie and I would go out into the field weekly. Every day we would go out and sample for crystal skipper numbers and record these numbers for our data. So we had transects at 8 total sites, as you can see here. This is an example of the transects that we walked at one of the sites at Fort Macon. So like the vegetation samples, we positioned random coordinates to form transects that that were across the plot. And as you can see, we kind of positioned them vertically and horizontally along the plot to maximize our surveying efforts. And we also positioned them so that when we were walking from one to another, we wouldn't cross across any of the transects to disturb any of the results from walking them. The methodology we used was the Pollard transect method. So this is basically walking a transect and counting observed crystal skippers along the way. So you can count the skippers by imagining a visual box about 10 to 15 feet to your left and right, above you, and then in front of you to count how many crystal skippers are present. So during this practice and methodology, it is assumed that you aren't going to count the same butterfly twice because it is kind of difficult to keep up with that as there are a lot flying around and they're honestly really fast and kind of hard to see. So while Katie and I would do this, we would switch off positions, but one of us would be the observer and they would have the GPS and walk in the front. So they would pretty much be the counter, counting all the crystal skippers that they saw and other insects that we saw as well. There were a lot of grasshoppers. And then the other one was behind and they were recording all that the person in front was saying. They weren't observing anything, just to keep everything consistent. And so we could accurately count how many that we saw. So this graph is the total numbers from both flight periods from 2021. So as you can see, Site B at Fort Macon had the greatest abundance. However, overall, we realized that the largest number in total that we saw was about 18 there, which is not a lot to combine both seasons. So this seemed to be a trend overall, as we didn't see any skippers at all at the skate park, Emerald Isle, or Salted Path or Bear Island. So this is really interesting because we were going out a lot during the week, almost every day, walking these transects multiple times. So it was really interesting that there weren't a lot of skippers that we saw despite the surveying effort. So as the sites were all along the coast, some of them were kind of far apart. And so we kind of wanted to look at areas in between these locations that had prime habitat for the crystal skippers just to see if there's anything there. So we made point counts at these locations that we found. And so at these point counts, we would be stationary and sit for a certain period of time to see how many skippers that we could see. Also, we would go to sites that we did have transects at and simply just walk the plot to see if we could find any there. And so when we weren't walking transects, this gave us more time to stop. And as you can see in the picture to the left, to get the perfect angle to get pictures of the crystal skippers. So what you see is Katie laying on the ground and I'm holding her hat with my bamboo stick to sift through the Seaside little Bluestem, holding her hat with that so it doesn't fall in the way for camera. So these are the final numbers for the point counts. So as you can see, there's a morning glory patch at Fort Macon State Park that had the most abundance of the crystal skippers there. Every time we would go, there were always a lot of crystal skippers flying around, so we would stand there and count them. And we also got to see a lot of times the skippers sitting in the flowers and crawling inside of them, which was really cool to watch. And so as you can see, for some reason, the point count numbers, we saw a lot more skippers than on the transects, which was really interesting because we didn't conduct the point count surveys quite as much of the transects, but these numbers got up to about 80 skippers, as you can see at the morning glory patch. So that was really interesting to see there. So this is just one last picture of me and Katie in the dunes. This was just a great opportunity for us to get to learn more about the native plants and animals that were in the dunes and about the crystal skipper. There's a lot of unknowns as to why there are so few skippers on our transects, on the point counts, whether this is related to the habitat or whether, but we're excited to keep analyzing the data and figure out more that we can find out. So at this point, I'm going to hand it back over to Dr. Price.
All right. Can you see my slides again? Just double checking.
Looks like we're seeing presenter view though.
Okay. Let me see if I can, here we go. There we go. Okay, that should be all set now. Okay,
Perfect.
So thank you, Maggie and Katie. I just want to give a shout out to these two ladies. I could not have asked for more hardworking students. If you've ever spent any time in the dunes in the middle of summer, it is pretty miserable. There are prickly pears. It is hot. There is no breeze whatsoever. And these ladies, sometimes it was raining and they went out week after week after week and always with a good attitude, even when they weren't seeing butterflies, which I know is very frustrating. So I was very proud of all the hard work that they did. And so in trying to ask, well, what do we do with all this data now? That's the important thing. So we've been collecting data and information on this species, but what's the point? What do we do with it? So the other thing that we have been working on is with the team that I have around me is a conservation plan for the species. So other species of butterfly have conservation or recovery plans that are put together to help have sort of a collective approach so that we can manage habitats and populations to support the species. And so that's what we're working on right now for the crystal skipper. It is not designated as a threatened or endangered species. However, just the fact that it is an endemic species that occurs over such a small range, we know that it's going to need some help to make sure that it persists. And so the conservation plan that we are working on is really fairly simple. The idea is to keep track of the crystal skipper population so we can continue to do population monitoring, point counts, and transect counts to be able to estimate what is the population size, where is it most abundant, look at occupancy. Is it staying in the same places all, from year to year? Or are they moving around? And then also being able to identify those key habitats, those stepping stone habitats, and assessing the quality of that habitat for the skippers. We're also interested in applying some more rigorous analysis as well. So using GIS approaches to use aerial imagery to identify places where we have very suitable habitat that we know, habitat that is completely unsuitable. So if there's a house or a condo or a parking lot, we know that's not going to be suitable skipper habitat. But what we can use this kind of information for is looking for those opportunity areas. So places where we do have good habitat that can either is already in good enough shape, or maybe with just a little bit of habitat management and restoration that doesn't have conflicting human factors, and maybe identify those as places where additional stepping stones can be either established or make sure that they stay in good shape. And we want to continue working with lots of partners, especially landowners. A lot of the beachfront habitat we think is probably in front of people's homes. So this is the restoration site there on Emerald Isle, and you can see where we've put in those plugs of Seaside little bluestem. This is good for the butterfly. It creates egg habitat, food for the caterpillars, but for the homeowners, it's also good because it helps to stabilize the dunes. So especially when we've had storm events that have sort of washed out those native plants, we can go in and replace that. And so it's an easy win-win, I think, for the landowners as well as people who are trying to help out the crystal skipper. So remember, the subtitle of this talk was How Does a Fish Biologist End Up Studying a Butterfly? And keeping this progress moving forward. And this slide is the answer to that question. It's because I can't do this work by myself. My job is to help motivate people and pull together experts and really motivated young people and bright students. But really, it's the experts from the natural resource agencies that you see here, other researchers at other institutions, Sea Grant, and so forth, that really make this a successful conservation effort. It really does take a team. And so I'm really excited about this work that we're doing. We have great momentum for this and great support going forward. And with that, we can take any questions.
Excellent presentation, everybody. I would encourage all of our viewers to give a great big virtual round of applause. Wherever you're sitting, put your hands together watching the program. Let everybody around you know you just watched a cool program. Or you can also drop like clapping hands emojis in the chat box. That works too. Carol, Katie, Maggie, thanks so much. Excellent stuff. And I do have some questions that are populating here in the chat. So if you're ready, I'll start throwing those your way.
We're ready.
All right. The first one that came up a little bit earlier in the program was, did you do any outreach to encourage private landowners to plant natives, or will that be in the future? And I'm curious about this too, the relationship. You had the locals there on your last slide. You mentioned at the very beginning that the crystal skipper is a little bit of a local celebrity. So I am curious about the local connection to the butterfly and to the habitats in your work.
Sure. So we have several sites that are privately owned, and the people that I've been working with and that Dr. Leidner before me worked with were fantastic about just giving us access to their property. So just allowing us out there to see if there's skippers around. They've been just fantastically cooperative. And every time we're out in the dunes and we're doing our surveys, people walk by. Homeowners walk from their top of their balconies down and come in and go, so what are y'all doing out there? And as soon as we tell them the story, they're like, well, you can come and plant on my property too. If you need a place to study butterflies, come over here. I've got them here too. And so it just seems to be a story and a species that sort of captures the imagination a little bit. And it's just wonderful to see that. And our original plan was to conduct, and this was working with Gloria through the Coastal Landscapes Initiative. We actually had in our proposal to go out and do meetings, in-person, presentations to the garden clubs and the, local community clubs around the banks. And because of the pandemic, we were not allowed to actually complete that part of our project. So it's still very much something that we're interested in doing. Of course, we also need to partner with nurseries so that if people want to have those plants available, they can actually go somewhere and find them to plant in their yards. So it's probably going to be before the end of this COVID situation before we can pick that back up. But we're ready to go. That will probably maybe be another great intern project.
Yeah, something to keep it going, right? All right. The next one comes from Jeff Hall. Any ideas why no skippers at Bear Island? It seems like lots of good habitat there.
Hi, Jeff. It's good to, well, I can't see you, but it's good to hear a question from you. Sorry, that's my cat Zoom bombing. So, we don't know. We were really discouraged about this. And I have to say, we scratched our heads, but and the answer is we don't know. We have no idea. Bear Island has not been surveyed for years because it was closed. It sustained a lot of damage from Florence. And so there was not even any access out there because it's already, you can only get out there by ferry and things like that. So it hadn't been sampled in a while. And I mean, we were out there, I say we, Katie and Maggie were out there every single week looking for butterflies. I mean, we scoured it. If there had been a butterfly out there, because they didn't just go to those sites, they also walked through the dunes, just to make sure that we weren't missing them. We also heard at Fort Macon, the numbers were really low. And so we feel like it was probably just a low year for this butterfly specifically. We don't know why. It could just be an anomaly. It could be sort of a sine wave kind of thing where this was just a low year and we might go out next year and there's just butterflies everywhere. We don't know is the answer. And but fortunately, I've added another partner to the team, Dr. Elsie Youngstead at NC State University. And she's, yes, you're nodding, Chris. It sounds like you might know her.
I know that name, yeah.
Yeah, so I was really excited to connect with her. I've needed a proper entomologist on this project for a long time, and I found one. And so I'm really looking forward with her to dive into some of these, to some of this data and see if we can come up with anything and maybe compare it to what people are seeing with other butterfly surveys, other species also in the coastal areas to see if this meshes with what they saw.
Excellent stuff. Exciting. Okay, let's see what's next here. Katie and Maggie, what was your favorite part of this research experience?
I'll go first. Honestly, it was just really cool. I've never done research before until I went to CMAST. And so just learning this whole process and getting to meet a lot of professionals in the field. And I never worked with butterflies before or knew a lot about plants. So I just feel like I gained a lot of knowledge that I didn't know. And so that was really cool. And it was really fun to work with Katie. She's a great person and just be out in the dunes every day. So I can't complain being outside.
Yeah, I definitely agree with everything Maggie said. I really enjoyed being able to feel like I was making a difference, in helping kind of conserving this endangered species and rebuilding its habitat. And I also just really enjoyed all the connections that I made. I've made friendships that I'm probably going to keep for a lifetime. And just the location we were at was spectacular. Also, we got to be at the beach every day. So that was really fun.
Yeah, I was gonna say, you can also say that the best part was beach ice cream at the end of a survey.
For sure. We definitely did that a couple times.
I remember I saw that last picture holding the popsicles.
Yeah.
I mean, yeah, it sounds like it would be it's a great place to do some research because the Carolina coast is just gorgeous. and you would get to learn about plants and animals that you wouldn't be learning about otherwise, because they do just grow in those habitats. So if you are a little bit of a science nerd, then yeah, it's going to be a lot of fun. That sounds exciting.
Was for sure.
Which leads actually into another question. Did you find many invasive plants or animals in your surveys?
Well, we actually we did. We found there was centipede grass actually growing in the dunes at some of the locations, which has kind of like, it's not an invasive species regularly, but in the dunes it is, because it's starting to encroach on the native species that actually grow in the dunes. So that was a concern that we were seeing. And there was another plant that we saw at Baptist Site, which was one of the locations at Indian Beach. And I can't remember the name of it, but it had like blue flowers, and it was really like not a good species to have because it kind of like drowned out all the other plants.
Beach vitex is that plant, which is also an invasive species in the dunes, and it just grows super, super fast. And the flowers are really pretty, which is probably why people have planted it there, is because it is really pretty. But unfortunately, it is considered an invasive species and not great for the native dune habitat. But we also saw lots of other cool critters. So, when you're out in the dunes, you see a lot of, snakes and lizards. And as Maggie mentioned, more grasshoppers than you can count. As you're walking through, they jump up and literally hit you in the face. So you're hot, you're sweaty. It's 100 degrees outside. You've got prickly pears poking you, and then a big grasshopper comes and like jams you in the face. And I think sometimes you just gotta wonder, like, maybe a desk job might be pretty good, but then you get one of those beautiful days where it's just glorious and you can go for a swim afterwards, and you think, yeah, this is the best job in the world.
As long as it's a desk job, like at a museum or with the Department of Environmental Quality or at NC State.
Yeah.
One of those are the good ones. Okay, let's see. I've got one more for you. And this kind of leads into that stepping stone idea for habitat. John writes, do we know how far these skippers will fly to find nectar? Any thought to use portable bait stations like pots of lantana to attract them as a survey technique also?
So those are really good questions. Some of that is answered in Dr. Leidner's dissertation and publications, because that stepping stone work was really with her. So she has that sort of critical distance you need between the stepping stones. And I'm sorry, I don't have that like right off the top of my head. But I guess her take home message was that for now, it seems to be sufficient. I think the next task is really to figure out where all of those stepping stones are, because we don't really know where all of them are. We just know where the big sort of critical ones are, but really doing that complete survey work. Now that is way more than any two interns could accomplish over, a single semester or even in a summer. It takes a big team to do that. So we're hoping that by having this conservation plan in place, that puts us in a position to apply for funding that can really bring a much bigger team, lots more woman power to this. In terms of the bait stations, I think that's a great idea. There's so many different ways to monitor populations and assess them. So that's again why I'm really glad to have experts like Dr. Youngstead to help us, and Dr. Nick Haddad and Heather, who can really help us, once we're moving into these more complicated, getting more in depth into this, figuring out what kind of approaches make the most sense to give us the information that we need to be able to communicate to private landowners. What can you do to be able to work with towns and municipalities to say, here's a little project that we can do that can have a big impact? Because that, I think, is really the key, is having the locals, having our local community understand how cool it is to have this little skipper in the dunes, and to keep them around for generations and generations.
I love that. That's fantastic. Dr. Price, Katie, Maggie, thank you so much for being on the program today.
Thank you for having us.
Thank you so
here.
much for having us.
Yeah, thanks, Chris. It's been really great. I really appreciate the invitation.
And I think we'll keep an eye on this work, and maybe next time we all find ourselves down at the coast again, keep an eye out if you happen to be near Bogue Banks for a crystal skipper. That'll be a fun one to look for.
Yeah, absolutely. If you see something you think is a skipper, pull out your phone, take a picture, put that into iNaturalist, and see if what you saw is one of our skippers, and we'll keep track of them with that platform for sure.
Excellent stuff. Well, everybody, there's another great program for you. Thanks for tuning in to this week's edition of the Lunchtime Discovery series. I hope that I'll see you again next Wednesday for another program. I remember next Wednesday we will be here. The following Wednesday we will not be here. So this is the last Lunchtime Discovery program for November. But yeah, we'll be back in December with another great lineup. Make sure that you're following the Office of Environmental Education on Twitter. They're at North Carolina EE, or head to their website, eenorthcarolina.org, in order to see the lineup for future presentations. All that information is also available at the museum's website, naturalsciences.org. And of course, you can follow us on social media. We're at Natural Sciences on Facebook, Twitter, and Instagram. And you're here on the YouTube channel. You might as well like and subscribe below. That way you can get notified when we bring you more great live programming. Like a show that's going to be starting here in just a few minutes right here on the museum's YouTube channel to help us celebrate our Darwin Days celebration that's happening virtually today, all day long on the museum's YouTube channel. And we'll be in person out on the Bicentennial Plaza outdoors with exhibits, activities, and crafts for the whole family all day Saturday, or 10 to 3 this coming Saturday. So I hope I'll see everybody in some of our virtual programs, and definitely this Saturday for Darwin Days. Until then, though, take care, stay safe, and keep your community safe. We'll see you again soon. Bye, everybody.